Well, aloha everyone, and welcome back to Anatomy 2 here at Chaminade University. Today we are going to be discussing the lymphatic system and its role in immunity. So, the goals of this chapter are for you to understand the lymphatic system in terms of both its structure and its function, and be able to compare the two different types of our immune systems, namely the innate and the adaptive immune systems. And then we'll talk about cell-mediated versus antibody-mediated immunity, which are going to be conferred by the T cells and the B cells. And then we'll talk about the effects of stress and aging on the immune response. So let's go ahead and dive right in. We have two major types of immune resistance. We have innate immunity, which is a non-specific resistance, and adaptive immunity, which is immunity that you acquire over time. So non-specific immunity or innate immunity is the kind of immunity that's present when you're born. This includes all of your natural defense mechanisms that are going to be barrier mechanisms or mucous mechanisms, and I think that's going to provide a general protection against invasion by a multitude of pathogens. And then adaptive immunity is specified immunity. It's an activated pathway where specific lymphocytes are available to combat particular pathogens or any other foreign invading substances. And the body system that carries out all of these immune responses is known as the lymphatic system. So the lymphatic system runs almost everywhere the circulatory system runs and has multiple different structures and organs that contain many different types of tissue, including lymphatic tissue, bone marrow, and a fluid which is called lymph or lymphatic fluid, and that flows within the lymphatic vessels. As I mentioned, the lymphatic system runs almost everywhere the blood does. So you see it in the marrow, you see it running up and down the limbs, you see it in the trunk, in the head, and then of course split, similarly to what we see with the iliac split. What's different though is that these guys are going to be single-ended vessels that are going to be similar to venous return in that they have one-way valves and they're going to work by drainage, and then they're going to be forced into, however, so instead of the way that the venous return does, we do have some filtration with the blood before it goes back, but the lymphatic fluid is going to be specially filtered through these things called lymph nodes. So you can see we have lymph nodes. We find them in our armpits. We find them in our throat. So when you feel like, oh, my throat's so swollen, I feel sick, that's because you have these submandibular nodes and the cervical nodes that are going to be what swells when you feel sick, right? We also have nodes again in our armpits and in our inguinal canal. And sometimes in a very common symptom of something called cat scratch fever, which is when we get infected, or toxoplasmosis, our blood gets infected by a bacteria that's very commonly found in cat fecal matter. So people who change cat litter boxes might get scratched by a cat that has been scratching the cat litter, but I digress. The point is that oftentimes the symptom of that is that they're going to have swollen armpits, and that's because these guys here end up overactive because they have a lot of toxin to combat, and so they end up swelling and swelling and swelling. So swollen lymph nodes is going to be something that's going to be a sign or symptom of an infection. Okay, so let's move on. What is the point of the lymphatic system? The lymphatic system serves to drain your interstitial fluid. So as I mentioned, all of the fluid balance in your body is going to come from the leaky capillaries leaking into the interstitial fluid. Then the interstitial fluid has an exchange across the cell membrane, where we have gas exchange and waste exchange. Then we're going to have that interstitial fluid then exchange back with the blood supply. But it also, in order to maintain that blood fluid balance, is going to leak into the lymphatic system. And so the lymphatic system serves to drain that interstitial fluid as well as to transport dietary fats. You may recall when we talked about digestion that the dietary fats are unable to be picked up directly into the bloodstream because they are what's called hydrophobic, and the bloodstream is aqueous or hydrophilic, and so we need a specialty carrier molecule to transport those dietary fats. So they're going to get transported through the lymphatic system and then get deposited back into the bloodstream that way. And last but not least, the lymphatic system is going to carry out all of the immune responses in the body. So we'll talk about those three major functions in a bit, but first let's talk about the architecture. So as I mentioned, lymphatic vessels start with closed ends. They're located in between the cells of the tissues and in the area where the interstitial fluid is going to be, and they're going to start merging to make larger vessels. And just like the venous return, we're going to see many valves, so one-way valves that are going to keep this fluid going forward. But unlike venous return, we're going to see all of these vessels merge into areas where we might have lymph nodes, or what we'll talk about later, called MALT or mucosa-associated lymphatic tissue. My point is that this pathway services to funnel all of the fluid that's picked up from the interstitial fluid through a mechanism that's going to help filter it, where it's going to run past things like macrophages, which are going to be digesting anything that might be found in that fluid. But I digress. We'll get to that in a second. We're continuing on with the architecture here. So as I mentioned, we have these one-way system where the fluid's going to leak in. It does that by going through these little openings between the cells. And that lymph is then going to travel through the lymphatic system. We're eventually going to get into lymphatic vessels and then funnel it through lymph nodes. And it's going to typically be found in between areas where we're going to have leakage, like the capillary bed. So this is the arterial. Remember, we have arteries, arterioles, capillaries, then venules, then veins. So here's the arterioles and the venules. We're going to have leakage of the capillary bed in between. And in that area, we're going to be picking up some of the interstitial fluid back into the veins, like we talked about with venous return, but some of it's going to get picked up here in lymph. And once it gets in, it doesn't go out until it gets funneled past something like the lymph nodes. All right. Apologize here. So from those vessels, the lymph passes through lymph nodes, and I mentioned the lymph nodes are going to serve us to help degrade and digest and attack any foreign bodies that might be in there that might come from invading microbes. And then eventually it'll feed into the lymph trunks. And we have a couple major lymph trunks. And again, these are going to run similar to where we see our vasculature, right? We have the lumbar lymph trunk, the intestinal, the bronchomediastinal, the subclavian, and the jugular trunks. And eventually the lymph trunks are going to merge to form two major ducts. The thoracic duct, which is actually the left lymphatic duct, or the right lymphatic duct. And then those are going to funnel into the blood supply. And that happens here. So here are our major ducts. Here it's going to go into, come on mouse, there we go. Right into here, the jugular trunk is going to feed directly into here's the right lymphatic duct. Here's the left lymphatic duct here. It's also known as the thoracic duct. And while it appears that they feed in evenly, that's not actually true. In fact, it's about 3/4 to 1/4. So the right lymphatic duct is going to drain the right upper half of your body, including your right arm, and half of your head and a bit of your abdomen. The rest of it, the other 3/4 of your body, drains in through the thoracic duct or the left lymphatic duct right here. Either way, they're then going to empty into the vena cava and directly into the right atria of the heart to go into circulation. All right, so this is just going to be a little bit of this is that box that we just saw here, but it's enlarged a bit. And this is showing you the routes of drainage. So here's our internal jugular vein, right? Here's our right subclavian vein. And just at that juncture, we're going to end up with the lymphatic duct. Here's the right lymphatic duct. On this side, here's the left internal jugular vein and the left subclavian vein. Again, at that juncture, we're going to have the thoracic duct, right? And either way, they're both going to feed into that vena cava, the superior vena cava, and then down into the right atria to start blood flow. So this is going to be an overview of the flow of the lymphatic fluid as relative to systemic and pulmonary circulation. So this diagram here should look, without the green, should look kind of familiar. Here's the heart, right? We've already talked about circulation. And remember, part of circulation is that we're going to go out to the lungs for gas exchange and then return back to the heart. So that's called pulmonary circulation. We also have systemic circulation, where this is going to represent blood flow to all of the rest of the body. And anywhere that we have a capillary bed, we are also going to have lymphatic capillaries that run right along the systemic blood capillaries, and then are going to get funneled into lymph nodes. They're funneled in by these afferent vessels. Afferent just means leading into, and vessels would be like the larger vasculature, except in this case it's lymphatic tissue. And this lymphatic vessels is going to feed into the afferent lymphatic vessels are going to feed into the lymph nodes, where we're going to have filtration of the lymph, and anything that's going to be in there, like foreign substances, etc., is going to both get filtered out and phagocytosed, as well as subjected to immune reactions. And we haven't talked about that yet, but we have B cells and T cells that both have specific immune function, right? Acquired immunity. And they're going to also be present in these lymph nodes and be responsible for setting up a systemic attack on anything that is an invader that they have seen before. After we've filtered up that lymphatic fluid, again, we'll have done that by way of phagocytosis and those immune responses. Then it's going to head into the afferent vessels. Again, we have a ton of one-way valves. You'll see one-way valves this entire way, just like we did in venous return, and that's going to help us ensure the one-way flow of lymph. Eventually, that's going to feed into the lymphatic ducts, and those lymphatic ducts are going to feed into the vasculature, right? And it's going to happen right at that superior vena cava, at the thoracic duct and the right lymphatic duct. And again, that's going to feed directly into the right atria and then head right on into circulation. So basically the flow of the lymph is going to be that it starts as the interstitial fluid and heads into those lymph capillaries. That's depicted here. And then it goes through the lymphatic vessels. Again, afferent into the lymph node and efferent leading away from the lymph node. All right, it's going to go through those lymph nodes and then out into the lymphatic ducts and then into the subclavian veins, which then head into the superior vena cava and into the right atria for circulation. Now we have other lymphatic organs, including the red bone marrow and the thymus. And we talked about T cells and B cells previously. This is where they get their names. T for thymus, B for bone marrow. And these specialty lymphatic organs are areas where immune cells become what's called immunocompetent. They go from being precursor cells to cells that are actually able to perform their immune function. Now, when you say thymus, you say, I'm not sure exactly where the thymus is located, unless obviously you've been listening to my other earlier lectures when we talked about endocrine system. But anyway, here's the thymus. The thymus is going to be located just beneath the throat and in between the two lobes of the lungs. This, of course, is going to be the fibrous pericardium. It's going to be the outer sheath that surrounds the heart itself. And so that's where the thymus is going to be located. Here's the thyroid gland, which is going to be found up near that hyoid bone in the throat, just all for reference. And then again, this is going to be where we're going to be dumping in our lymphatic fluid through those lymphatic ducts. Okay, so let's look at the thymus. Here we have what's called a Hassel's corpuscle, or a thymic corpuscle. That's going to be this area that's in the region in the center here. It's called the medullary region, or the medulla. So as we've seen before, the cortex is the outer section. So as you're heading inward, you're going to go through the cortex and then enter into the medulla. But the first thing that you're going to hit is going to be trabeculae. All right, so here we have a capsule. The capsule is the entire, going to go around the entire thymus. Directly underneath that, we have that subcapsular space that's going to lead us directly into the trabeculae. Right, from there we're going to filter through the cortex and then into the medulla. And again, if we're going to take a detail of that medulla, here we have an up-close vision of thymic or Hassel's corpuscle. We also have other tissues like lymph nodes, which I mentioned, they're going to serve to help filter the lymphatic fluid. We have the spleen, and we'll talk about that in a second, but it's going to be a region where we're filtering the blood, similar to what we do with filtering the lymph nodes. And we have lymphatic nodules, which are going to be similar to lymph nodes, but they're not associated with these particular lymphatic pathways, and more found in our mucosal tissue. So they're called mucosal associated, so MALT tissue, mucosal associated lymphatic tissue. All right, but I digress. Let's talk about a lymph node. As I mentioned, fluid comes in through the afferent lymphatic vessels, so it looks like this, right? As it heads in, it's going to first enter into the subcapsular sinus. So here's the capsule. The subcapsular sinus is going to just be this region here. Then it's going to enter into these trabeculae. These trabeculae are going to be like openings or gaps in between, where that fluid is then going to end up filtered inward through the cortex and then into the medulla, right? In that inner medullary region is where we're going to end up having a lot of the digestion. So inside each of these regions, we have specialty cells. I haven't really talked about these specialty cells or what they do, so let's focus on that for a second, and then we'll talk about where they reside. So we have B cells and T cells. These are both involved in acquired immunity, right? We also have dendritic cells and macrophages. These are going to be involved in innate immunity. They're going to be eating pretty non-specifically. The T cells and the B cells differ in that the T cells actually go out and do the dirty work, and that means that they actually are the ones that go out and fight the invading cells. The B cells, however, are going to create what's called antibodies, and antibodies are going to exit into circulation and going to target invading cells for degradation, but the B cells don't actually degrade them themselves. All right, so in each of these regions, you can see that we have different combinations of these cells that are meant to filter and degrade anything in the lymphatic fluid. Inside the outer cortex, we're going to have B cells. In the germinal center of the outer cortex, we're going to have B cells as well as dendritic cells and macrophages. These guys are going to be pretty nonspecific. Remember, they're going to be chewing up anything they can find. It's going to be the process of what's called phagocytosis, where they have these little pseudopods that are going to reach out and engulf anything that they're going to want to eat. They basically invaginate that, and then they wrap all the way around. It becomes inside. They internalize it, and then they degrade it by way of lysozymes. I mentioned B cells make antibodies that are going to help mount an immune response, but they themselves are not the soldiers. T cells, on the other hand, are going to be specific, and they do work with that similar antigen reaction. However, they themselves are going to have receptors on the surface that are going to bind directly to the cells of interest and target them for degradation. In any way, pretty much nothing stands a chance. Once it comes through these afferent lymphatic vessels, it's going to encounter all of these cells before they leave the afferent lymphatic vessels, which means that by the time the fluid leaves its afferent lymphatic vessels, there's pretty much nothing left that is non-self. Everything has been degraded by these cells that are pretty much just sitting here being fed a buffet of any sort of foreign invaders that have entered into your body. So again, the route of lymphatic fluid through a lymph node means that we're headed in through that afferent lymphatic vessel shown here on the left, into the trabecular sinus, which is this area here. Once we're in that white area, we're going to go into the medullary sinus and then out through the afferent lymphatic vessel, right? And each of these different areas is going to have specialty cells that are going to degrade whatever is coming through that lymphatic fluid, such that when it leaves by that afferent lymphatic vessel, it is clean. So again, here's a structure of that lymph node. Here's what a lymph node looks like. As you can see, it's very tightly attached to the surrounding areas, and that's because it's being fed by these afferent lymphatic vessels. Inside the lymph node, the fluid that comes in through that afferent lymphatic vessels is going to enter into the subcapsular sinus, then into the trabeculae sinus. And then we'll go ahead through the inner cortex and into the medulla, the medullary sinus, and then it'll head out through those afferent lymphatic vessels. In this way, it's going to be subjected to multiple different sets of cells that are going to clean everything that comes through. So when the time it leaves in this efferent lymphatic vessels, it's clean and can enter back into the blood supply. Now the spleen is going to do something similar. However, the spleen is going to work to filter everything that's found in your blood supply as opposed to in your lymphatic fluid. So here's what the spleen looks like. The spleen has an impression both from your kidney, the renal impression, and the end of your stomach, the gastric impression. So the reason that these dents are here is just because other organs need to fit around them. We're going to have vasculature that's going to be pretty intense. So we have the splenic artery that feeds in, and it's going to then have filtration into multiple different tinier arteries. The splenic artery splits, and then each of these are going to enter into what's called the pulp region. So we have the red pulp and the white pulp. The red pulp is going to be blood tissue, right? We have venous sinuses where we're going to have leakage of blood into this region, this yellow region. See, we also have these trabeculae. We're going to have something very similar occurring here. We have a bunch of T cells and B cells that are macrophages and dendritic cells that are going to be attacking anything that's in that blood supply that is non-self. And then the clean fluid is going to get picked up and sent out through the splenic vein. So in this way, the vasculature is also going to get filtered through lymphatic cells like T cells and B cells by way of the spleen. But the spleen also services not only to take away non-self, such as foreign invaders, but also to degrade any sort of red blood cells that have reached the end of their lifespan, or platelets, or any sort of cells that have gotten to the point where they are ready for degradation, which really means recycling, right? We want to make sure that we can utilize all of the parts of these cells. When we break them down, we're going to use them to make new cells. And so that's also going to be the purpose of the spleen as well. All right, so MALT tissue, or mucosa-associated lymphatic tissue. These are lymphatic nodules that are not surrounded by that capsule that we saw when we talked about the lymph nodes. So a little bit more free floating or free moving, and they're scattered throughout the mucous membranes, specifically throughout the lamina propria of those mucous membranes that are lining all sorts of different things, including your gastrointestinal tract, your reproductive tract, your urinary tract, and your respiratory tract. And think about it, these are all going to be areas where we have exposure to the outside, whether we've eaten something, right, or it's actually a tube that's connected to the outside, like our urinary tube, right, or a reproductive tract, again, a tube that's connected directly to the outside. Respiratory airways, we're actually breathing in air from the outside, which might have foreign matter in it. So these are all going to be first lines of defense for regions that are going to be subjected to outside invaders. And again, these are referred to as mucosa-associated lymphatic tissue, or MALT tissue. This diagram is not going to be tested. It talks about the development of lymphatic tissues. Just briefly, they develop from these sac regions. Here's the jugular lymph sac at the back of the neck, the retroperitoneal lymph sac, and the posterior lymph sac, and then these are going to then reach outward and then eventually become all of the lymphatic tissue. And this occurs as the circulatory system is developing as well. So these two are going to develop in concert, again, such that the circulatory system is always going to be able to deliver stuff to the interstitial fluid, which is then going to be picked up both by the circulatory system return through the venous return, but also through this lymphatic tissue. So again, they're going to be co-developing during development. Okay, so we've talked about general immunity. Let's talk about innate immunity. I kind of touched on this a little bit earlier, but let's talk about the specific types of innate immunity that we have. The two major lines of defense include our skin and our mucous membranes. Anything that's going to be an internal defense as well, like our chemical types of, like our stomach has low pH, etc. So our major types of defense are going to be either physical barriers or chemical warfare, such as a low pH. Or also, we have enzymes, for example, in our tears. So anything that's going to be flushing out as well has chemical defenses. So our mechanical defenses, as I mentioned, include our skin. It's going to be our very first barrier function. Also mucous membranes and tears, or liquid from those mucous membranes, including tears, saliva, mucus, etc. Again, all of this is going to be served to help push things outward, which might help to flush out or directly keep away through mechanical defenses like the skin, foreign invaders. We also have cilia, which serve to move fluid along. We have that in our trachea and our reproductive tract, etc. We have our epiglottis, which serves to make sure that when we swallow, food goes one direction, but when we're breathing, air goes another. We also have urine flow, so one of the best lines of defense for an urinary tract infection would be to drink a lot of water, which means that we're going to have high volumes of fluid, and bacteria aren't able to hold onto the wall when they first come in and to start an infection, and they'll just be flushed out. And if you're able to keep your urine flow high enough, you're able to prevent that infection from taking hold. This is not going to factor in if we have later on stages of some sort of infection, because bacteria make like a capsule, and all of a sudden the urine flow doesn't have as much of a role. However, urine pH does have a role in breaking that down, and we'll talk about pH when we talk about chemical defenses. Other mechanical defenses, again, are going to include fluid things like vomit or defecation, etc. Chemical defenses include anything that's going to be high or low pH, so gastric juice, also anything that's going to be enzymatic like lysozymes, sebum, etc. Some of our internal defenses include things that are phagocytes, anything that are going to actually phagocytose foreign matter, like our macrophages and our dendritic cells. We also have natural killer cells. Natural killer cells are going to serve to roam through our system and attack self that is abnormal self, such as cancer cells, etc. We have the inflammatory response, whereby we can call macrophages over to a site that has also had blood vessels added to it, and we are generally going to have an increase in temperature at that location, or we can have a global increase in temperature in something like a fever. All of these serve to hopefully kill off, and if not kill off, at least lower the reproductive span or reproductive rate of things like bacteria and other foreign microbes, such that our body's immune system is able to actually attack them so that we can maintain homeostasis. All right, so what does phagocytosis look like? Phagocytosis is when we're going to have these little pseudopods. Those are here. The pseudopods reach out and engulf different microbes. How do they know where to go? Well, there's something called chemotaxis, and that's going to be a chemical signal that a microbe is going to secrete. Might be byproducts of its metabolism or something like that. It's going to be something that's going to be detected by this phagocyte that's going to actually be a migrating cell. It migrates through the interstitial fluid until it finds the microbe. Once it does that, again, the pseudopods reach out and engulf the microbe. The microbe becomes adhered to the inside of these little pseudopod regions and eventually engulfed in a phagosome, which is the process of ingestion. and then it's going to be digested by way of a lysosome adhering to that phagosome. The lysosome is going to have digestive enzymes in there. Those digestive enzymes, when combined with this foreign body, are going to break it down into things that we can hopefully reuse. Anything that can get recycled is going to remain or get exported for the ability to be transformed into whatever it's going to make. So building blocks are going to be then sent out to other cells or kept within the cell for its own metabolism. And then whatever is unable to be digested would be a residual body, and that's going to be secreted and then popped back out into the interstitial fluid. Now, this is the process of inflammation. Now, inflammation relies on a couple of things. One, we have to have chemotaxis. Basically, that's going to be something released by the microbe that's going to call the phagocytes over. We're also going to have vasodilation and permeability of the vasculature. All of this is going to happen by particular molecules, we'll talk about that in a minute, in response to tissue injury. So this represents, you cut yourself on a knife while cooking, or you stepped on a splinter or something. And typically, what you cut yourself with, unless it is a scalpel and it is in an actual operating room, more than likely it's not sterile. And that means that you've introduced some sort of foreign body, these microbes, into the underlying tissue. And when that happens, those microbes, again, they release some sort of byproducts of their metabolism, which are going to then be detected by the phagocytes via chemotaxis. But what also happens is that macrophages have a special signal that they can release that calls for more macrophages. So this one can say, hey, this is more than I'm able to eat. More of you need to come out. And that's a process called emigration. And emigration occurs both by chemical signals that are going to be coming from the phagocytes, but also changes in this vasculature that allow these cells, which were normally wandering through the vasculature, to actually emigrate out of the blood vessels to be able to go to the buffet and digest the microbes. So this diagram depicts phagocytes migrating away from the bloodstream into the site of tissue injury in order to help with tissue repair. Okay, so we have many different lines of defense. Again, we talked about the skin and the mucous membranes as our first line of our defense. The physical factors, again, include the epidermis of the skin. That's a physical barrier to the microbes getting in. We also have mucous membranes. They're also going to be a physical barrier. We have mucus, which serves to trap microbes in things like the respiratory and gastrointestinal tracts. And then these can be eliminated in the form of sneezing, right, or blowing your nose. You can then eliminate it. We also have hairs that are found in our nose and in other regions, but in our ears, for example, they're going to help filter out microbes and also dust. Additionally, we have cilia. Cilia are these little regions on top of the cell that are going to help move fluid along the surface of those cells or the cell layer. And they work in concert with mucus, and they're going to be moving the mucus along to help trap and remove microbes and dust from that upper respiratory tract. Additionally, we have the lacrimal apparatus, which is our tear ducts. Tears are going to serve to dilute and wash away irritating substances and microbes. They also have little enzymes in there that can help degrade things. So we're going to have a little bit of chemical factors in there. Saliva is going to serve to help wash microbes away from the surface of our teeth. Also the mucous membranes of our mouth. Urine is going to hopefully help wash microbes from our urethra, help avoid a urinary tract infection. And the purpose of defecation and vomiting is also to help expel microbes from the body as well. So if you get very sick, you can end up with diarrhea or excessive defecation or also vomiting. This is also going to result in extreme fluid loss. So you're going to want to make sure that you're keeping up with your fluids if you're experiencing those symptoms. Chemical factors are going to include things like sebum. Sebum is acidic. It forms a nice acidic film over our skin surface that helps prevent the growth of different microbes. Helps us from getting skin surface infections. We also have lysosomes, which are antimicrobial substances. These are found in a lot of those fluids that I talked about earlier, like the saliva and our tears. Found in sweat, tears, saliva, our nasal secretions, and many of our tissue fluids. Additionally, gastric juice, really low pH, helps destroy bacteria and most of the toxins that enter into our digestive pathways by way of the stomach. And again, we're also going to have a region that's going to have contact with the external environment, namely the vagina. So we have specific secretions in the vagina that are slightly acidic to help discourage bacterial growth and flush microbes out that might come from an external entrance. Some of our second lines of defense include our internal defenses. We have specialty antimicrobial substances such as interferons. Interferons are going to help protect uninfected host cells from viral infection. Basically, cells that are infected are going to release signals or upregulate these interferon signals, which are going to mean that the surrounding cells, which are yet uninfected, can increase their defenses such that we're able to help retain uninfected host cells. We also have a complement system, and we'll talk about that in a little bit more detail, which causes cytolysis, or breaking down of the microbes. Also helps promote phagocytosis, or the specialty cells which are going to phagocytose foreign invaders. And it's going to help contribute to inflammation. Now again, the inflammatory response serves to help kill off, or at the very minimum, reduce the reproductive capacity of the bacteria or microbes in that area, such that and it also brings additional things like macrophages, etc., over, such that our immune system can help attack whatever it is that has come to infect our system. Specialty proteins that are iron binding proteins are going to be upregulated, and these are kind of neat. Basically, these guys inhibit bacterial growth by reducing the iron that's available. We know the iron is needed for the bacteria to undergo replication. And so if we can bind up all of the iron in the surrounding region, that bacteria is going to be slowed in its replicative capacity, which means that our immune system might have a chance to get a handle on the situation. We also have specialty natural killer cells. These guys work by releasing something called porphyrin and granzymes. Both of these are going to have different methods that are going to provide the same function. They're going to help have the cells basically implode. They're going to have little holes that are formed in their surface, which are then going to allow phagocytes to help kill any of the microbes that are internal that are going to be released. Phagocytes are going to be specialty types of cells that are going to ingest any type of particulate matter that's considered non-self or foreign. And again, inflammation is going to provide that inflammatory response that going to help to confine the microbes, destroy the microbes, and then eventually initiate tissue repair. This is a localized response, whereas fever is a global response. And it's going to help increase the overall temperature. This is going to help inhibit microbial growth, intensify the effects of those interferons that we talked about, which helps the uninfected cells remain uninfected, and speed up repair reactions within the body. All right. Now, adaptive immunity, I talked about it a little bit previously. We're going to dive into it in a little more depth here. This is the specialty immunity whereby the body is able to defend itself against specific invading agents. It does that by the antigen antibody response. Now, antigens are going to be substances that are recognized as foreign that provide immune responses, and antibodies are going to be specialty types of not cells, but biomolecules that are released from cells that are going to seek out particular antigens. And this happens kind of by like shotgun science. So we have a ton of different, know the expression, a million monkeys and a million typewriters will eventually type the works of Shakespeare. And that's kind of how this works in your body too. We have a lot of different cells that are randomly making these antibodies or presenting parts that are going to be found on the T cells, and they're going to randomly either encounter or not encounter the antigens in the foreign matter. And once we do have one that works, we're going to hold on to that in our memory bank such that the next time we encounter that foreign body, we're able to mount an immune response more quickly. Again, adaptive immunity has both specificity, as I mentioned, is going to have an antigen antibody response, and also memory. This is going to allow us to be able to mount an immune response more quickly that second time that we see our foreign invader. And it's divided into two major types, cell-mediated and antibody-mediated. It's also divided along two specified cell lines. So cell-mediated is going to be the T cells, and antibody-mediated is going to be B cells. All right, so again, the T cells come from the thymus and the B cells come from the bone marrow, and they're both going to have precursor cells that are going to become mature cells. So the precursor cells are going to be the pre-T cells that become the T cells if we're talking the thymus. And the mature B cells are going to come from the red bone marrow. Both of these cells can enter into circulation. This is why we find them in the spleen, right? We also find them in the lymph nodes. We find them in the malt tissue. They can also just be found circulating through an attempt to find any foreign matter that might be as yet undetected. Either way, these mature cells are going to find their final location and then serve to attack specific foreign invaders that are going to match up with their receptors. So both the T cells and the B cells have antigen receptors, the difference being that the T cells keep the antigen receptors on the surface of their cells, whereas the B cells secrete those in the form of antibodies, which then go into circulation and bind to and inactivate antigens that are going to be found on foreign invader cells. All right, so T cells are going to have to undergo the formation of helper T cells, which come from these helper T cell precursors going into these helper T cell clones. Anytime a helper T cell clone is going to create an active T cell that's going to be effective, we're going to create a memory of those helper cells. And the same thing is going to happen with these cytotoxic T cells. If we find one that is effective, then we're going to create a memory of it. So these memory cytotoxic T cells are going to lie in wait so that the next time we don't have to enter through this whole pathway, we can just go right through here, the activation, and then enter into circulation. So that's going to allow us to mount an immune response much more quickly the second time that we encounter a foreign invader. Again, the major difference between T cells and B cells is that T cells are the soldiers. T cells with those active antigen receptors on their surface are actually going to go out and have cell-mediated immunity. It's a directed immunity against intracellular pathogens, also sometimes against cancer cells, and additionally, unfortunately, against tissue transplant cells. Can also happen, an immune rejection response can also occur against a fetus, for example. This is why people who have tissue transplants need to take immunorejection drugs, and why women who are pregnant end up with a lowered immune response. This is why they also can't eat things like soft cheeses, which might include microbes that normally the body would be able to attack, but because they have a lower immune response, they're unable to attack it and can get sick. So again, this is all due to what's called cell-mediated immunity, which occurs through those T cells. Now, B cells work a little bit differently. B cells are going to maintain in the plasma and then create these antibodies. These plasma cells are going to secrete antibodies, which are then going to circulate out to find antigens and target cells that have those antigens for destruction. Again, anytime we have a B cell that's particularly effective, we're going to make a memory of these B cells, and those memory cells are going to serve to help bypass this whole pathway such that we can just zoop into the creation of those antibodies and secretion of those antibodies such that we're able to mount our immune response more quickly the second time that we see a foreign invader. All right, so all of this works by way of immunogenicity and reactivity. What does that mean? We have to have something on the surface of the cell that has what we call an epitope. We'll talk about that in a second, but we're going to call it an antigen. And that antigen is going to be what's detected by an antibodies, which causes what we call reactivity. Now, we can react with the entire microbe, or more likely just parts of the microbe may serve as what we call antigens, and we can typically have multiple different regions of the antigen that are going to be detected that can trigger an immune response. Those small parts of the antigens are called epitopes. And what the immune response does is it mounts by shotgun science multiple different versions of these different biomolecules that may or may not see epitopes. If the epitopes are present, then we are able to mount that immune response, and we're going to save the T cell or B cell that creates it in the form of a memory cell such that we're able to mount that immune response more quickly the second time. So this would be the entire antigen, the entire region that can be detected by way of antibodies, but typically only a small region of the antigen, i.e. an epitope, is going to be detected by the antibody. Now, in order for that immune response to occur, either the B cell or the T cell must recognize that that antigen is present, and in order to recognize that antigen, we have to recognize an epitope. And in order to recognize it, we have to have a specialty processing that occurs. Now, B cells are a little bit different from T cells in that they are going to secrete those antibodies, right? And they're able to recognize and bind to antigens in the lymphatic tissue, in the interstitial fluid, or in the blood plasma. But T cells, these foreign soldiers, they're not quite as smart. They can only recognize fragments of these proteins that are processed and presented in a particular way. And that is going to be this macrohistocompatibility complex, or an MHC complex. So what happens? The MHC complex is created once the antigen and antibody interact, and then that's going to get created and inserted into the plasma membrane of the body cell. It's going to get presented in a particular way that the T cells are able to detect it. It's called antigen presentation, and that looks kind of like this. So here's our exogenous antigen. That antigen is going to get phagocytosed, Once it gets phagocytosed, then it's going to get digested into smaller peptide fragments. These peptide fragments are then going to get broken down and then presented in these specialty MHC2, this is the MHC2 molecules, into a vesicle that's then going to get presented on the outside of the cell. So here's our MHC molecule, that self-antigen, and here's the peptide fragment that's going to get presented on the outside of that cell. All right, so here is another version of this endogenous antigen processing. This occurs with MHC1 self-antigen, but it works in a very similar way. Basically what happens is, again, we have that endogenous antigen as opposed to what was an exogenous antigen. So this was going to be something that was external and is going to be picked up by the cell. This is something that is internal, that is going to then be presented on the outside of the cell. And that's going to happen when body cells are infected. And this is going to be kind of like an SOS, which basically says I'm infected. And either it does a couple of things. One, it's going to help the surrounding cells lock their doors, if you will, to prevent them from becoming infected. And two, it's going to target itself for degradation. So this would be if we have something that's occurring, like, for example, a virus has infected the cell. So this is an internal way by which the cells can say SOS. So again, we have that antigen. This time it's endogenous. It's going to get broken down into different peptide fragments and processed similarly inside this, this vesicle, which is then going to get packaged with the MHC1 complex internally, which then gets popped externally. Here's the MHC1 with the antigen presented. And this is going to be inserted directly into the plasma membrane of an infected body cell to be able to say, hey, I'm infected, both A, save yourself, and B, come help me attack my foreign attackers, or undergo what's called apoptosis so that all of what can be salvaged of the cell can be recycled. And this occurs by way of multiple different types of protein hormones. Okay, so we have a lot of different protein hormones that are involved in the immune response in many different ways. These are called cytokines. One of the cytokines is going to be interleukin 1 or IL-1. We also have IL-2 and IL-4, IL-5, IL-6. But all of these interleukins are produced by either macrophages or helper T cells. And their goal is to stimulate the proliferation of T cells or B cells such that we can secrete antibodies or activate natural killer cells or cause a fever. All of these are going to be involved in the immune response, right? We also have tumor necrosis factor, or TNF. This is produced by macrophages. And this is basically when I said previously that macrophages might need help, they might call in other macrophages because the buffet is too large for them to eat. That's TNF. TNF stimulates the accumulation of different neutrophils and macrophages at the site of inflammation. And again, it's released by macrophages. I need more macrophages, calling all hands on deck. And that's going to help stimulate them to kill the microbes in that area. Interferons are specialty molecules that are created by cells that are infected by viruses to help A, inhibit the replication of the virus in uninfected cells, so telling those cells to lock their doors. Also to activate cytotoxic T cells to help come and help attack them because they've been infected. And natural killer cells, again, natural killer cells are going to attack self that is under attack, things that are under viral attack, for example, and help inhibit cell division and suppress tumor formation. Additionally, we have macrophage migrating inhibiting factor. And this is basically going to prevent macrophages from leaving the site of infection. So they're going to be drawn there by TNF, and they're going to stay there by MMIF, which is produced by cytotoxic T cells. And basically, it's going to say you will stay here until we have digested every single one of the microbes that have infected this tissue. So that's the role of cytokines in the immune response. And I'm not going to ask you which of the specialty cytokines are involved in which ones, but I do need you to understand the role of cytokines in the immune response, namely what are the major steps that occur in the immune response. And remember, these are all going to be happening kind of simultaneously, or at least all overlapping. Okay, so I talked about cell-mediated immunity a little bit previously, in that the T cells are going to recognize antigens that are presented on the surface of foreign invaders And the T cells are then going to bind that antigen and eliminate that antigen. And we talked about that happening by way of shotgun science, basically, shotgun immunity. We are going to create a ton of different types of T cells that all randomly have different recognition capabilities, and the ones that do recognize that antigen are going to differentiate into a clone of effector cells. So all of a sudden we find out that you're effective, we're going to upregulate the amount of you that is circulating, the amount of those T cells that is circulating, and that's going to result in the elimination of that antigen. At that same time, not only are we going to upregulate the amount of active T cells, we're also going to upregulate or save some of these T cells in our memory pile. These are going to be much longer lived than our active T cells, which are eventually going to die off after the infection has been cleared. But these memory cells are going to perpetuate for a long period of time, which means that the next time that this foreign invader is present, we don't have to activate this co-stimulation pathway, etc., whereby MHC2 is activated when we have the receptor on that cell is presenting that antigen, or is going to react with the antigen that's presented, by the MHC complex. The MHC complex is presenting the antigen, it's reacting by that TCR, and that's going to cause the linkage of the CD4 protein, which causes activation of the helper T cell. So that's the details of what happens in that pathway that I just gave you the overview of. But my point is that that takes a little while. Whereas here, if we have these memory helper T cells already, we can bypass that simply by entering into proliferation and differentiation. So we just kind of shoop right in here, which means that we can speed up the immune response the next time that we see that foreign invader. All right, now here's a cytotoxic T cell clone, which is similar to what we just saw previously, except these were helper T cell clones. The major difference being which type of histocompatibility complex is going to be presenting itself. So the MHC2 is going to be for an exogenous antigen. Remember, MHC1 is going to be for an endogenous antigen. So this is why it's going to be cytotoxic. That means it's going to be killing self, but it's going to be killing infected self, so it's okay. That's also going to happen by way of CD8 binding to the MHC once the TCR, or that receptor that's going to be able to bind to that antigen, is going to be able to have actually bound the antigen. That's going to cause the stimulation of an inactive cytotoxic T cell to become active. And just like we saw previously with the helper T cells, the cytotoxic T cell is going to undergo a series of clonal selection, then proliferation and differentiation. And that means that the ones that are going to be active are the ones that are going to have actually been formed to demonstrate to be effective. And additionally, we're going to have created those memory cytotoxic T cells at the same time, right? Two different pathways, which means that we're able to use those memory cytotoxic T cells much later on even to be able to bypass that whole activation pathway, which again is going to allow us for a faster immune response, right? All we have to do is come out of memory and enter into proliferation and differentiation. All right, so how do these cells work? All right, so here's an activated cytotoxic T cell, and it can do two different things. It can either use granzymes to attack this infected body cell, which means that the cell walls itself, or cell membrane itself, I apologize, this is an animal cell, so no cell wall, but the cell membrane is going to end up with holes in it, which means that that cell is going to undergo apoptosis, and there's going to be phagocytes that are drawn to that general area, such as macrophages, etc., that are going to destroy any of the microbes that are released, and anything that's going to be able to be recycled is going to be recycled. There is a little bit of a difference over here from a different method that the cytotoxic T cells can take, whereby we are going to secrete granulysin and perforin. Perforin puts holes and makes channels, which is going to allow the granulysin to come in. The granulysin is not only going to cause apoptosis of the actual infected body cell, but also cytolysis of, this is apoptosis or programmed cell death. This is cytolysis or cell bursting. But my point is that this occurs not only for the infected body cell, as well as it also is going to affect all of the microbes that are in there. So there's no need for the phagocyte to be present. The microbes are destroyed directly by the granulysin. And both of these pathways is going to result in the destruction of the infected body cell, but also result in the elimination of the microbe that has caused the infection. Okay, so next we're going to talk about antibody-mediated immunity, in which an antigen is recognized and bound by an antibody. Helper T cells are going to serve to stimulate the B cell Now the B cell is going to be, again, a bone marrow cell, so that the B cell can proliferate and differentiate into a clone of multiple different types of effector cells that are going to produce antibodies. And again, this is going to happen by way of shotgun. So we're going to make a ton of different effector cells that all produce different antibodies. And then eventually one of those antibodies is going to work. The one that is going to work is going to then be proliferated. And therefore, we're going to have a ton of the effective ones, which means we're going to have an increase in the antibodies circulating through, which targets the cells for destruction, eventually leading to the elimination of that antigen. So that works kind of like this. So we have an inactive B cell, which has B cell receptors, which are basically precursors to antibodies. These B cell receptors are then going to be able to become, they're going to interact with microbes. Note that they're interacting with different regions of different microbes, which causes the ability to, by way of shotgun, have some of them recognize an unprocessed antigen, one we haven't seen before. Now, once we recognize it, we end up with co-stimulation. Again, that occurs by way of the helper T cells. And then we are going to end up with clonal selection of these activated B cells, which results in, again, the memory cells, which are much longer lived, and the plasma cells, which are going to end up secreting antibodies. Antibodies are then going to end up interacting with our target cells that are found on the microbes, or the different epitopes that are found on the microbes, which are the target cells, I should say, which targets them for destruction. Again, these memory cells are going to serve to help bypass this whole pathway the next time we see that foreign invader, such that we're able to more quickly mount an immune response. All right, so let's look at antibodies. The most common antibody that we're going to find is the IgG. And the IgG basically looks like a Y, like a fancy Y with two heavy chains, and they're called heavy chains because they're longer, which means they weigh more. We also are going to have some disulfide bonds here that hold them in place, and some specialty carbohydrates that are found here, but we have a hinge region that makes it look like a Y. On the outside, we have these light chains, light because they're smaller and therefore don't weigh as much, and these are what are going to be different, okay? These regions here, these variable regions, are going to be these V regions on both the inside of the heavy chain and the outside on that light chain are going to be the regions that are going to see the antigen. So these are going to be the binding regions. Now, I just described IgG, which looks like a Y, but we also have several others, including IgD and IgE, which also look like Ys, kind of. These are very low concentration in the blood, 0.3% for both of them combined. IgG, however, most abundant, about 80% of the antibodies found in blood. We also have IgA, which looks like an IgG, but kind of duplicated, so we have barbell style instead. That's about 10 to 15% of the antibodies, and IgMs, which are going to look like snowflakes. As you might imagine, IgAs and IgMs have more regions to be able to interact with the antigens. But generally, the IgMs are going to be found as monomers on the surface of these cells where they serve as receptors. So they're not typically going to be circulating. We do sometimes have circulating some that are going to be found in the plasma, though, and these are going to be what are going to go awry in an incompatible blood transfusion. So if we put the wrong blood type from a donor into a recipient, we can end up with clotting reactions, or agglutination reactions, I should say. It's not actually a clotting pathway, but it causes blood clots, which will lead to the death of the individual. This is why they're so cautious when giving blood transfusions, to make sure that they match the blood type specifically. All right, so let's talk about the complement activation, which I talked about previously. Let me first talk about the goal of this so that it helps us keep in mind when we talk about what's happening to think about why it's happening. And the goal of complement activation is cytolysis, or the creation of holes in the channels of the membrane of a microbe such that the microbe bursts due to too much extracellular fluid flowing in. And that happens because we have these channels that are created by C9, as well as a little complex of C6, C7, C8, and C5b. Okay, so all of these different proteins have to come together to form this specialty channel, which is going to allow what's called cytolysis, or the breaking of the microbe down because too much fluid has come in. All right, so what happens? We are going to end up with phagocytosis. So phagocytosis is going to be enhanced by coating with C3b. All right, so the first thing that has to happen is C3 is going to get split into C3b and C3a. C3b is going to get coated on top of the microbe to help the microbe be phagocytose most effectively. C3A is going to join with C5. Again, C5 is going to split into C5A and C5B. C5A and C3A are going to join together in the mast cell to help increase histamine and upregulate inflammation or our immune response. Now, when you want to take an anti-inflammatory, right, sometimes you might take an NSAID, but sometimes you might take an antihistamine like Claritin or something like that. That's because histamines are going to cause inflammation, which is going to result in a couple of things. One, extravasation of microbes from the blood vessels because of increased blood vessel permeability, and also chemotactic attraction. So remember, this phagocytosis is going to occur, which is going to it's going to happen because the microbes are going to say, hey, I have something in my chemicals, right, something of my metabolism. It's going to be released into that fluid, that interstitial fluid, causing these phagocytes to come over, right? That's called chemotactic attraction. All of that is going to be increased by histamine, again, because of C3A and C5A coming together. But what happens to C5B, C6, C7, C8, and C9? Well, exactly what I told you at the beginning. They form this complex which causes cytolysis of that microbe, which is going to occur because of these molecules aligning, right? So we have a lot of fluid that's going to come in from the extracellular fluid and cause this cell to burst. That is all the process of complement activation. All right, this slide serves to talk about immunological memory. So on the left, we have the primary response, which will be the first exposure to some sort of microbe. And on the right, we have the secondary response. So I'd like you to compare the response in both the IgM and the IgG. So the IgM is going to be the first thing that's going to mount a response. It's going to be the blood plasma, and it's going to be also found mounted to the B cells, right? And we're going to have a spike in our antibody about a week later. We also are going to have a response in terms of our IgG. Again, we're going to have an increase, so we have a titer of about 1, and this is an arbitrary unit, but here my point is that first time, if it's considered 1, the second exposure not only happens earlier, but it's going to have about 100-fold, 500-fold it looks like, somewhere around 300 to 500-fold increase in the antibody titer of IgGs in our second response. It happens faster, it happens stronger, and typically it knocks this microbe out before you even know that microbe is there. Typically this results in no actual response to the organism because they don't feel sick at all because we've eliminated the microbe pretty early on. You'll note that IgM response is pretty much the same as what we saw the first time, but that IgG response is out of the park. All right, so let's talk about adaptive immunity. Again, adaptive immunity is immunity that you acquire over time. And when we think about that, we typically think about naturally acquired active immunity. Basically, you are exposed to a microbe. We end up with recognition by B cells and T cells, which forms those specialty T cells, and also a specialty T and B cells, and also forms those specialty memory cells, which can mount our secondary immune response the next time that we see that foreign microbe. But that's not the only way that we can get active immunity, right? Or we can also acquire adaptive immunity. I'm sorry, I said active. I meant adaptive. It's not the only way that we can get adaptive immunity. We can also acquire it passively through natural ways, such as getting it from the mother to fetus across the placenta, or by drinking it through the breast milk. We can also get IgA antibodies and IgG antibodies from our mothers. And that's naturally acquired, but it's considered passive. Now, we can also have artificially acquired, both active and passive. And artificially acquired means that we mount that immune response by way of introducing something. In this case, this is our vaccination event. So when we get a vaccination, and I want to be clear here that the COVID vaccine doesn't work this way. The COVID vaccine is an mRNA vaccine, which works in a totally different way and is for a different class. But most of the vaccinations that you've gotten in your lifetime are going to basically happen when we have given either a weakened version of the virus, or generally at this point it's not even a full virus, it's just parts of that virus that are going to be introduced to the body in the form that's an antigen. And that's going to stimulate that cell-mediated and antibody-mediated immune response without ever actually seeing that virus, which produces memory cells so that if you do see the virus in the future, you can bypass all of the early stages and go directly to attacking that foreign body. Now, antigens are pre-treated. That means that they're going to be immunogenic but not pathogenic, which means that they will trigger an immune response but not cause a significant illness. What do I mean by significant illness? Well, pathogenic antibodies would be something that would be the actual virus itself, a native virus. But our antigens are going to be something that I mentioned are pre-treated, either just little small parts of the virus or weakened versions of the virus so that it can cause the immune response but not any actual significant illness. And last but not least, we can have artificially acquired passive immunity. And this is very relevant right now. This is going to be when we take a blood transplant or transfusion, from an individual who has already seen the pathogen. For example, COVID-19. We're using this regularly to treat people who are extremely sick, which is why they're asking for plasma donations from individuals who have been confirmed to have had COVID and have been recovered from it, because they are thought to have, they are known to have antibodies in their system that are able to help mount an immune response. And if we put that blood in an individual who is the same blood type but is suffering from that virus, we can give them artificially acquired passive immunity such that they are going to have antibodies that can help mount an immune response and hopefully help stave off the severity of their disease. All right, so how do we select for T cells? T cells must be able to self-recognize but also be self-tolerant. So they have two major factors, self-recognition and self-tolerance. So they undergo two different sets of selective pressure, a positive pressure to ensure that they are able to recognize self-MHC antigens, but also a negative selection to mean that they will not react with other self-proteins. Again, this is known as self-tolerance, and if this goes wrong, we enter into some autoimmune disease disorders such as lupus, for example, or arthritis. All right, so we have a couple different steps in this pathway. We have an immature T cell, and the first question is, does it recognize self-MHC proteins? And it has to, or else it's not functional. So if it doesn't, we're going to induce it to die. So that's our first selection. It's a positive selection, I suppose, because the negative cells are going to die off. That's a positive selection. So now that means that the cells that do recognize self-MHC proteins are going to move on to bind to and recognize self-peptides. This is going to be a negative selective pressure, which means, that the ones that are going to undergo this choice and then decide that they do recognize self-peptides and bind to those self-peptides are either undergoing deletion or death, or what's called anergy or inactivation. And so this is a negative selective process because we don't want the T cell to have receptors capable of binding to and recognizing self-peptides. In this factor, we have a positive selection for those that are going to recognize self-MHC and a negative selection for those that are not going to bind to and recognize self-peptides. After this, we also are going to have T cells as they emerge from the thymus in the lymphatic tissue. So this is the mature T cells. So mature T cells are going to undergo activation via co-stimulation. So if we have antigen recognition with that co-stimulation, that means that we're going to enter into proliferation and differentiation of that T cell. If we have antigen recognition without co-stimulation, that means that it's going to be aberrant, and we're going to enter into the inactivation or anergy of that T cell. We could also have a deletion signal occurring, which leads to the death of the T cell. All right, so in this way, again, we have self-recognition and self-tolerance. Now, B cells also develop tolerance, and again, they're going to do that through deletion and anergy. So here's the pathway that B cells are going to undergo. Does the immature B cell recognize self-MHC molecules or other self-antigens? If the answer is yes, then we're going to have a deletion or death of that B cell in the bone marrow. If the answer is no, so again, this is negative selection, we do not want it to recognize self-MHC or other self-antigens, right? It's going to be a negative selection. Now, if the mature B cell recognizes the antigen under the first signal, that means co-stimulation is going to occur. We'll have the co-stimulation with the second signal. That's going to cause the activation of the B cell. If we don't end up with co-stimulation, that causes anergy or inactivation of the B cell. And again, this occurs in our secondary lymphatic tissues and in our blood. And that's going to lead to anergy, again, or inactivation, because we want to undergo such that the cell is The immature B cell is unable to recognize self-MHC molecules. The mature B cell is able to recognize antigen and cause co-stimulation, which is going to lead to the activation of the B cell. In this way, B cells are also going to develop both self-recognition and self-tolerance. Now, this is a summary of the different cells that are going to be present and responsive for the adaptive immune response. That's going to include our antigen-presenting cells, such as our macrophages, our dendritic cells, and our B cells, as well as lymphocytes, such as our cytotoxic T cells, helper T cells, and our memory cells. All right, so macrophages are going to be responsible for processing and the presentation of the foreign antigens to T cells. Remember, T cells are soldier cells, and they're kind of thick-headed. They need to have that MHC complex with the antigen presented in a particular way on a silver platter for them to be able to detect it. Macrophages also are going to secrete IL-1, which is going to secrete IL-2 by helper T cells, and induce the proliferation of B cells. The secretion of interferons are also going to happen from these macrophages, and interferons are going to stimulate T cell growth. Dendritic cells are going to process the antigens and present the antigens to T cells and B cells. These dendritic cells are found in mucous membranes, skin, and lymph nodes. And B cells are going to help process and present antigens to helper T cells. Lymphocytes are going to include things like cytotoxic T cells, which kill our host target cells by releasing granzymes. Remember, granzymes are going to help induce apoptosis. Also going to release perforin. Perforin causes channels. Those channels cause cytolysis. And that specialty granulysin that destroys the microbes by way of destroying the microbes themselves as well as the cells that they have infected. We also have a lymphotoxin. Lymphotoxin is going to help destroy the target cell's DNA. And cytotoxic T cells are also going to use gamma interferon that attracts macrophages. Macrophages are going to also, remember, they're phagocytes. They're also going to upregulate their phagocytotic capability with this gamma interferon. And macrophage migration is going to be inhibited by MMIF, which means that the macrophages are going to stay in place at the site of infection until they have consumed everything on the buffet, right? We have helper T cells, which cooperate with B cells to help create antibodies and amplify antibody production. Also help secrete IL-2 and help stimulate the proliferation of T cells and B cells. Helper T cells also secrete gamma interferon and tumor necrosis factor. Remember, TNF is going to be involved in macrophages calling other macrophages over to the site of infection. This is all going to help stimulate our inflammatory response.
Mom,
Hi, baby. B cells are going to differentiate into plasma cells, which are going to secrete specific antibodies. Remember, this happens by way of... You can go get them from the closet, sweetheart. Can you get juice from the closet for her right next to the fridge? I apologize, guys. I digress. We also have plasma cells. Plasma cells are descendants of B cells, Descendants of B cells that are going to help produce and secrete those antibodies. And then the memory B cells are going to be the descendants that remain to help be responsible for that secondary immune response, so we can respond rapidly and forcefully if we ever encounter the same antigen in the future. There's an entire field that looks at how stress is involved in the immune response, and that's psychoneuroimmunology, or PNI. And this field deals with common pathways that link the immune system with the nervous system and the endocrine system. And the current research in this field has demonstrated that your health levels and the disease state of your body is directly influenced in some parts by your thoughts, your feelings, your moods, and your belief systems. We also will look at aging in the immune system. And in terms of the immune system, aging results in increased susceptibility to infections and increased malignancies, as well as an increased production of autoantibodies and an decreased response to vaccinations. It also can result in decreased immune system function overall. This image talks about homeostatic imbalances, and we're going to specifically be talking about AIDS. Now, HIV is the virus, human immunodeficiency virus, that causes AIDS, or acquired immunodeficiency syndrome. And this is a condition when an individual is going to have a higher rate of infections because the immune system has been destroyed step by step by HIV, or the human immunodeficiency virus. And this just shows you what HIV looks like. It's an RNA virus which intercalates permanently into your DNA. It's called a retrovirus. And these are actually very dangerous because that means that every cell that has been infected will pass that infection on to its daughter and descendant cells. So there's really no way to clear yourself of the virus entirely once you've been infected with the virus. However, we've made great strides in terms of the pharmacology, and we're able to treat these individuals for, well, basically forever, so that they maintain their normal lifespan. So we talked about histamine and how histamine was involved in the immune response. Now, allergies can occur when a person is overly reactive to some substance that most people tolerate well. And typically that means that we're going to have an overreactivity in that histamine, which again, we talked about how that works and what it stimulates in terms of stimulating an increase in temperature in an area, etc. And if we have a sustained allergic reaction, typically this can result in tissue damage. And we have 4 main types of hypersensitivity, types 1 through 4, and we're not going to test you on this, so just wanted to let you know that there are different types of allergic sensitivity. Anytime that we have an immune system that is out of balance, where it fails to display the main hallmarks of the immune system, namely self-tolerance, that's going to mean that we're going to be able to attack the body's own tissues. This is an autoimmune system disorder, and again, I mentioned this would be something like lupus or arthritis. So that brings us to the end of our discussion about the lymphatic system. I really appreciate you taking the time to join us today. So thank you very much. Aloha and happy studying.